Hi guys, today I'm in cell membrane because we are going to talk about the transport across membranes, cellular membranes. Cell transport is the process of how things move in or out of the cell or the organelle through the cell membrane. There are two broad categories of cell transport: passive transport and active transport. It depends on the type of molecule that is going to be transported. When we talk about small molecules, passive transport. When we talk about large molecules, active transport. So if we start talking about passive transport, it's important that we know that the molecules are going to move in the favorable concentration and electrochemical gradients, from higher concentration to lower concentration. For that reason, energy is not necessary. When we talk about passive transport, means diffusion. We are going to review the meaning of diffusion. It's a diffusion process. There are different types of diffusion: the passive diffusion or simple diffusion and the facilitate diffusion. When we talk about passive or simple diffusion, means that the molecule can cross the lipid bilayer directly. They are very small molecules, because as you can see here in our picture, only small molecules and hydrophobic molecules can cross the lipid bilayer, but large polar molecules are not allowed to do that. Okay? Another important concept here is the osmosis. Osmosis is when we transport water across the lipid bilayer, across the membrane. In that case, the movement of water permits that we introduce a new term, tonicity. What is the meaning of tonicity? It's the ability of an extracellular solution to make water move into or out of the cell or organelle. Okay? It's possible that we talk about three different types of solution. Isotonic solution: in that case, our cell maintains the proper morphology, there's no change in the cell. Hypotonic solution: in that case, we are going to see that when the cell wants to maintain the equilibrium between outside and inside, we are going to observe how the cell is going to be larger. Okay? And there's the possibility that we observe an hypertonic solution. In that case, we are going to see how the cell shrink. Okay? It's going to be smaller. In these two cases, it's possible that our cell die. Okay? In that case, in osmosis, the water is going to cross the lipid bilayer directly. The other type of diffusion is the facilitate diffusion. In that case, we need proteins for transporting our molecules. There are two types of proteins: the channel proteins and the carrier proteins. When we talk about channel proteins, they are open pores. It's like a door that permits to cross out or in to the cell or to the organelle. Okay? There are specific pores, such as aquaporins, that permits the movement of water. So we talked before about osmosis, and now we are talking about aquaporins. In osmosis, water crosses directly the lipid bilayer. Here they are using channel proteins. Well, in this case, when they use channel proteins, the movement of water is quicker than when they move by osmosis. The other type of channel proteins are the ion channels. Ion channels are very important because they permit very quick transportation of molecules. Okay, but in that case, the selectivity is higher than before. Okay, these channels are not open all the time. We require a specific situations for opening them. So that permits to classify these channels into two groups: ligand gate channels and voltage gate channels. When we talk about ligand gate channels, means that we need a specific molecule, the ligand, that bind to our protein and permits to open the door. This is our signal molecule, bind to this specific site and permits to open the door. And then molecule can cross. When we talk about voltage gate channels, this is very important for nervous system. Channels opens in response to change in electric potential across the membrane. Okay, as you can see here, we have the membrane the electric potential, and we are going to change this charge, and that's open the door and then permits to cross the molecule across the membrane. With the voltage, this change, the door is open and then the molecule can cross the membrane. The other type of proteins involved in facilitate diffusion are the carrier proteins. In carrier proteins, it is compulsory that our molecules bind with the carrier protein, with the transporter. Okay, if we compare carrier proteins with ion channels, in ion channels there's no junction between molecule and protein, but in carrier proteins it is compulsory that our molecule bind with the specific protein. So as you can see in this picture, not in this one, as you can see in the picture, this is the molecule that wants to move to the other side. This molecule binds to this area. This is the specific site. Okay, when that happens, the molecule binds here. We are going to observe a conformational change. That change permits to move, to travel the molecule to the other side. So this is the, in that case, in our example, we have this is the glucose. This is the glucose binding site. When the molecule binds to the specific site, there's a change of the conformation of these carrier proteins, and that permits the movement of the molecule to the other side. Okay, what's about active transport? Active transport means that molecules are going to move from lower concentration to higher concentration. That means that they are going to move against a concentration and electrochemical gradients. For that reason, energy is required, ATP is required. There are different types, okay, driving by ATP hydrolysis or co-transports. We can include co-transports here. Driving by ATP transporters, driving by ATP hydrolysis, here there's the ion pumps and ABC transporters. Ion pumps, the most famous ion pump is the sodium-potassium pump, in which each ATP molecule moves 3 molecules of sodium outside of the cell and permits the movement of 2 potassium inside, as you can see here in this picture. Okay, sorry. Oops. ATP, sodium move outside, potassium move inside. This is very important because maintain the osmotic balance and cell volume. Okay. The other type of active transport, ABC transporters, they are going to use ATP as well, and they are more focused on the movement of toxic molecules, toxic metabolites. Okay, so they can prevent some illness. Co-transports can be included in active or passive transport. Okay, the co-transports means that 2 molecules are going to move. Okay, one of them requires energy and the other one doesn't require energy. It's possible to talk about symport and antiport. The symport means that we are going to move 2 molecules in the same direction, in or out. Okay, antiport, these 2 molecules move in different ways. For example, the pink one is going to be inside and the green one moves outside. Okay, they are going to move in opposite directions. The term unipole means that a single substance moves in a single direction. Okay. When we talk about large molecules, we talk about macrotransports, and here there's three different types: endocytosis, exocytosis, and transcytosis. Endocytosis means inside. Okay, in that case, our cells or the structure is going to take the molecules from outside, the large molecules from outside, and they are going to move inside. That requires the formation of a vesicle, okay, and requires ATP. So, in that case, it's possible to talk about different types of endocytosis: phagocytosis and pinocytosis. Phagocytosis means eat. In that case, our cells is going to eat solid things. For example, another cell. Typical example, bacteria. A macrophage, a type of cell, eat a bacteria. Okay? When we talk about pinocytosis, means to drink. We are going to take some soluble molecules, and the cell is going to drink, or the organelle is going to drink that. Okay? When we talk about pinocytosis, it's very important that we consider that there are different types, and one of them that is very important is the endocytosis mediated by receptors. Okay? That is, in that case, it's important that the molecules that are going to be integrated in the cell will be recognized by a specific receptor. Okay, that's an important point. When we talk about exocytosis, in exocytosis ATP is required as well, and in that case we are going to expel substance from inside to outside. Requires vesicle formation as well. Finally, we have transcytosis. We are going to combine endocytosis and exocytosis. As you can see in this picture, we are going to take molecules from here, from the lumen, okay, by endocytosis. This vesicle is going to travel across the cell cytoplasms, and then molecules are going to be expelled to the other side by exocytosis. Well, that's all. We did a small review of the transport across plasma membrane. They are very, very important. We give only a general view because in physiology you are going to study them in depth. Okay, it's important that you check the videos that I'm sharing you. Okay, because you are going to see animations of these processes. You have this presentation in the Campus Virtual, so that means that you can check all the tests and add the things that I didn't mention here in that moment. Okay, because remember, this is a review. Well, don't forget to follow the updates in the Campus Virtual and review this material because it's very, very important, because we are going to do a specific assignment in class. Okay, see you.